single pha navegun nitak lama enti nitak pha previous class kha structure of pollen grain and microsporogenesis and the formation of the male gametophyte pollen grain kha ki zoma dunia kha female structure te ki pantadi as actual pollen grains are two male gametes ki mo ya male. So that is already done, which is ready for fertilization. So now today we are going to study the structure of pistil, ovule, Megasporangium lekichi, and then Embryos sec. Tomato nekizildimo. So it is some bangwaka, gynoecium a female part of the flower engine. Pistil or carpel lekichima hat pepe nazatheun. Pistil or carpel it consists of how many parts? 3 parts. Mala stigma, style, and ovary. I come I think muna banga ipoimo pino ichile ovary sunga XII on ki form de janga pin This part is very important. It is how it stigma a function, style a function, and so the stigma chubang sticky. Mala stigma is always sticky. As chubang, why sticky? Because they have to receive the pollen grains. Mala it helps in receiving pollen grains. Pollen grains they come. I want to stigma on to do, bang chidan de on to do like pollinating agents te tung tona ka. Mala pollinating agents like bees and different kinds of animals as well as human beings. Why te tung tona ka? Hay pollen grain te on to the pollen grain pollen tube ki form di nathila ya pollen tube form di mala. So stigma in pollen grains they can receive the engine style pai suk the engine style chu bang mahilo it is a tube shape tube shape hiloma tube mala style chu tube shape and it connects what stigma and ovary abana ovary chu chang it will contain asunga ovary chu it will later form what ovary arrive chang when it becomes ripe it will form the fruit, and inside the ovary banga ovulomage hayas hay sunga khama ovulomage hay ovulin it will form seed later on mala thil atang him to lai bang en leuchin hai tang chi khong thil tang him men leuchin ovulage ovule pen kha hon le changa khabang when it becomes mature it becomes seed. So we are going to study the structure of ovule first. What is the structure of ovule? And then ovule pen koi sunga omi chang ovary sunga ovary bang ovary when it becomes mature it ripen ovary is known as what? Fruit. And the matured ovule is known as seed. So we need to know the structure of ovule. Why ovule draw diwa what is the structure of ovule? Ovule it is also known as Megasporangium. As I told you, mega it's big, micro it's a Megasporangium. Sporangium tjanga sunga XII te hongome ichanga mole. So we're going to study the structure of what? Megasporangium. Structure it ngai jumale. Don en leutin. Hay ovary juma stigma style de drawn on lo. Hay ovulomage. Hay ovule sunga bagama che just study it wai bang kadi sim diu kha. Hay ovule to ovary ki akizop ngai male they have to correct somehow so why a connecting napin kha bang kichi funicle kichi delam chale chu draw thau di ma ai pin kha funicle kichi that is the joining part of the ovule and ovary male and then funicle kichi chin hai funicle pin kha hai to ki attach gi chi thei deu ma abana ovary sunga kha tun for example la kha funicle na themo funicle bang ovule to ovary ki attach na ma and then tun e banga orange kongen ta zwal na hol hai ipan masang mo orange kongen ta zwal ha asite bang chi oma male bang asite khong hile asite changa bang ovule hiloma bang ovule the structure itu male tampiom hai ovary sunga kha cavity om male a homo minchin hai pin kha ovarian cavity kichi why ovarian cavity chu bang lobule I just introducing you to some important terms which occurs inside the ovary mum avana hai kha I ovule here which will become the seed hai ovule pen to kha aki attach na ma haya si ovule te ki attach na pin hai pin khaba hai kha placenta kichi tun human a eleuchin le anaw pang naw ngek pin kha anu to anaw ngek ovary sunga kha uterus sunga kha banga anaw ngek pin to anu aki attach na om ve male agui te kha wai pin kha bang kichi wai structure kha placenta kichi chu ha so a placenta penin bang bolai chile anuin anek ting kha anawin lende here chidan de wai placenta in kha hatang pia kha an pia chu male food in chinwai jaga kha a placenta pin kha it is name cheu ma cushion dai sofa dan de tia kha ki thwai te dan kha ki line chen cushion like structure kichima placenta is a cushion like structure which is located inside the ovarian cavity have ovarian cavity pen bang kichi locule kichi malam hai te bang ovule kha will later become seeds so this khen khaten kha ovule tampini umo like seed khada you know for example ovule kha chawamble ovary khada one ovule in an ovary while ma for example a mango chini mango mala tura ya hai te bang tang khatom ai bana example dang wheat paddy chi khong le ma wheat and paddy while gelun la wheat and paddy abana atam de wa many ovules leom thema na many ovules here tang tampi om na mu here theina bang khadu che theina semu che waite banga example papaya khong singtang mai khong phel a leo chena sunga tang tampi om male here watermelon male chi khong park sahi park te orchids chi khong aza kha white to ki depend kha many ovules ovarian changa kha papaya watermelon and orchids ovule kha chom changa chu like mango wheat and paddy so this is the structure overall structure of the how it is attached how the ovule is attached inside the ovary so to kha main topic ovule pen kha structure kha hai diagram kha poimo nakizilgi ke te una siama tanu poimo structure diagram kha hon swak zale changa kha exam khonga kha ovary to draw se non lo ya aninga ka draw dia ovary sunga ovule koi sunga ovary sunga bang to ki attach funicle to ki attach male hai kha stalk chia so hai pen kha en ne wala ovule pen kha to kha lian de winom dejo hai kama ovule pen kha chin an oilam ki lang lo sial chia lang toi chin to kha ovule pen kha you know seed here amole ovule to seed in chin obviously they will have they'll have a hole or a pore somewhere right so that from those seeds why apara hon por khe deun chin banga thil on germinate thak zelu hi amole seed e changa kha hilo mamale wai kha hom na mun kha om te wai hom na mun pen koilam dia chile ule tung lama zong draw the ule noi lama le draw the mo so I'll draw this side will be my what micropyle kichi a homom da pen kha bang kichi my grow pile kichu he kha porom na a homom na him pen kha micropyle he bang jaga ki draw masai chi le micropyle to opposite and draw opposite a op na chet pen kha abang en kichi chalazal en kichi micropyle opposite a kha chalazal en kichi chet ma helama micropyle na draw le helam kha chalazal en hisel di opposite to micropyle kha chalazal en hi chet di ma abana hai ovule pen kha si jai changa kha ma kha covering nei du hilom ma for protection a kha why covering ti kha bang kichi ai chi le why ti kha integuments kichi draw mala integuments kichi second a draw dan mo step by step a kha hak san na sa kule kha integuments kichi chin ma hai kha integuments bang china cover tu china hele le chu mala wai za kha Megasporangium kha ovule kha integumented kichi integumented Megasporangium mo integumented ovule kichi integuments on zia ka khama ai kha second na poimo hinchin micropyle koilama khat pe pewa hom na mun pe pewa mo like a drona na hinchin hoi opposite abang om chalazal hai tegal chang ta mo abana coverings te kha bang kichi integuments kichi abana hai sunga kha unball na omzel ngai mo le ha sak cho sunga kha ana pya ngai we changa kha XII te ke hai unball na pen kha bang kichi nucellar region le kichi ma nucellus pen kha unball na pen kha hinchin aba ne hai sunga kha hai structure kha honom hai kha hai bang structure embryos so embryos kichi yak chu hai sunga hai kha embryo hon swag ve changa kha mo le embryos ka kham hai chu extra kha delam changa extra kha izil ngai du to hinai lo mo le ovule sunga me changa kha draw him ngai hai teng kha to ma kha izil ngai du hai kha XII we jo ya like mam pen ma XII hinchin hai embryosic abana so this is done chalazal nucellus micropylar and integuments mo le hai te kha structure we jo structure kha na draw zo changa kha a hi na auto function te kha abana gel zel di mom so it's done de hinchin abana poimo khada hile funicle pin kha ko yam ta le e funicle kha ovary to akizom na pen male funicle stalk pin kha elama ka draw to the funicle stalk kha ai kha stalk chu bang funicle chiangam funicle hi ta mole hai kha stalk hin chin hai pen kha hai funicle pen kha ki extend he chu funicle pen kha a stalk pen kha ki extend zel ma y extension pen kha bang ki chia hai la ki extend to mole ki extend to hai extension pen kha bang ki chia chi le extension pen kha rafi kichi hai te kha nawa de kha terms te kha thangen chin mo so the hoi dewa ngai deu rafi kichi bang kichi RAPHE rafi le kichi ma diagram le bang kha dom de textbook le om ni so what is the structure of ovule ma explain the structure of ovule chi le ma sa pena kha hai kha ovary to akiates namun pen kha bang kichi funicle kichi then a homom na pen kha micropylar en kichi opposite a kha chalazal en kichi a covering stick kha bang kha kem bit tu te kha integuments kichi an bol tu pen kha sunga om pen kha nucellus kichi abana extension of the micro extension of the funicle pen kha rafi ki chin abana haya enleuchin haya a body hai integuments ma of integuments to hai body pen to ki attach namun pen chet kha hilam kichi where it attached to the body hilam kichi. So these are the few important points in the structure of ovule. Ovule structure draw what it's a hai ting kha kim te te ngai mo diagram hoilo thoto ami tale a labeling pen kha hoi de wana gel ngai clear ngai funicle. So let's repeat what is funicle. That is a stalk, the stalk of an ovule. Stalk, this stalk STLK. Funicle is a stalk of an ovule. Bana micropylar is the pore where there is an opening. More opening of pore ha micropylar. Opposite to micropylar is what chalazal n. Hitchedi. Abana the coverings were known as integuments. Nucellus is present in the central portion, more where it supplies food and other processes. And then hilum pincha, the attachment to the body. More rafika, extension of funicle. And inside we have embryos sac. And embryos sac we are going to study in detail later. So here change in structure of ovule. Drawing la, label la bana. We can go to the next. Ovule khamom types tampio mention. For example, to aya pin kha textbook pin kha cha mejo ovule kha for hai lambang hai homom no pen bang kachidi micropylar en kachithia dikmale micropylar en opposite side pen bang hidi chalazal en male jin micropylar pen akha bangom di chalazal en hinchin bana holdile ha micropylar en chitale micropylar opposite abang chalazal abana enleu chin kha ovule kha to textbook am pen kha ovule pen khai cha kha khen khati khatang zen zan lo in chin ha ya pa a funicle pen kha mo cha kha on ki extension. This is the funicle male ha funicle bang hitale a position kha bang chia chi la kha inverted position now male chidan de inverted ovules de khama bang ki chai chile anatropous ovule ki amin ma anatropous ovule that's it tun for example khen khatte ovule khen khatte khama 90 degree kha tang zen zen amu micropylar en hai lambang hitale ya hom na male micropylar en bang hitale ya kha chalazal cha opposite a formula khana the ngai wai chu micropylar en na gel na na wa opposite chalazal hidi cha abana kha stalk le kha kitang vek thia micropylar chalazal to kha kitang vek ale om the chu itan da watanga mu changa bang ki chi dan de te tang zet ti kha bang ki chi orthotropous atang pia kha tamatam mo types chu le hai te gal cho le hun lwa mo ja chu textbook kha anatropous o mention wa adang te le gup lam khongom to mo om na position to am kha so a straight am changa kha ortho kha the exam le ortho to straight ma le thil himi ma straight atun for example in gu khong le kisu hekta le kha hon su tang tu pin kha banga ortho lam ma le ortho lam to chi changa ki sai changa bon lam hiloma so ortho kha straight am changa kha ortho ki chin inverted position a kha mo onom changa kha we call it anatropous ovule. This is just the position of these three with respect to these three funicle, chalazal and micropylar. So a textbook pen khamam anatropous ovule jo inverted ma. So that is what we drew. Textbook lekha eche ame chu consodi. Mole namu he ai kha micropylar cha micropylar hai micropyle cha micropyle opposite abang om chala zal pol ki chi mo opposite chada in chin hai khatang gem gam loi chang khai cha koi sime che kha abang ki chi anatropous ovule ki chi male anatropous ovule inverted position in chin hai lama ki draw ken chu den lama cha ka draw ki bang thoma hai lama draw le hai lama draw to le as long as the alignment is correct funicle pen khabang stalk right stalk of an ovule abana a body of the ovule kha funicle to aki attach na mun pen kha body attach na mun pen kha haya na mu ya hilum ma hilum hinchin abana outer and inner integuments these are the integuments khabanga covering cha nucellus pen kha haya unball na pen kha inchin nucellus sunga bang embryos ko male embryos sunga te kha atwa mazil kid so this is the structure of ovule to a structure of ovule kha kisota inchin to kha lexa kha bang chidan dewa embryosic on ki form here and XII tele khoya oma chikha and wala construct Megasporogenesis and mesanu banga Megasporogenesis Microsporogenesis na theilayo hiam why tole difference le khamo thengai jo Microsporogenesis kha bangchi pollen mother cell apada kha the formation of the microspores four microspores from the pollen mother cell by the process of meiosis is known as Microsporogenesis mala tole Megasporogenesis kha de la nucellus ka chena pen khan theima from the nucellus kha ama kha hon ki differentiate diun chin simple the draw tele hai kha a micro nucellus ita jo nucellus portion pe na khamam hon the differentiate diun chin differentiate changa hon ki wanga ki khen tuam diu cell khati ha ke nucellus amu non lo can cell dang ka form mu chidima nuclear region akha nucellus cells ken kha hon ki ovule songa kha micropylar region akha ma micropylar lama kha ati nucellus de kha ken khati hon ki khen tom diun chin hon ki differentiate diun chin kha cell kha hon form di wai kha bang ki chi megaspore mother cell pollen mother cell remember pollen mother cell dan de wa kha hele megaspore mother cell hon ki differentiate diun chin hai megaspore mother cell in kha cell kha de ta chuma differentiate zo no kha hai cell kha din kha cell ni hon form de in chin cell kha din kha wait I'll draw in a different color so that you will understand the cells hai kha megaspore mother cell chitani ma hai kha cell ni hon ki form ta cha hai cell ni hon ki form de in chin abana cell ni hai chu one apara tu hi le thai sama le bang hi di mitosis ma meiosis mitosis akha chin 1 2 3 4 cells on ki form dio which an ki form chin kha aya kha micropylar region male hailam kha here micropylar region excuse me you can don't put sorry aya kha micropylar region lama kha thum kha azan na di hon be di chele hoi zo lo ma set lam ma non chin hoi kha english power bang chi ai chele three cells degenerate ki R-A-T-E the three cells in the micropylar region will degenerate and only one will be functional te changa chuma banana sim theli ma cell tampi form thelai di chinai chu only one will become functional male one functional megaspore ki so the formation of the megaspores as a whole and by that we mean we got one functional megaspore so the formation of megaspores from where from the megaspore mother cell MMC lekichima from megaspore mother cell is known as Megasporogenesis. What is Megasporogenesis? The formation of the megaspores from the megaspore mother cell is called Megasporogenesis. We can write it down. The formation or the process of formation of megaspores from megaspore mother cell, MMC, is called Megasporogenesis. And this is a diagram. You can draw it. Megasporogenesis. Now what we have to do next, megaspore, functional megaspore in mga form to di mo so that we can get what the embryo sac then la mawala bol di ka chipin mo embryo sac pen ka bang chi form di ovule sunga ka bang jaga embryo sac form bang jaga embryo sac isilo chi chang embryo sac sunga XII di XII changa ha pollen grains de hon lo do changa ha fertilization the mo. That is why we have to go through this process. So what is Megasporogenesis? Okay, I think I'll erase now if you understand. Doon, hay functional megaspore but in continuing out the lay. We can call it how does the embryo sac develop? Development of a female gametophyte. Male gametophyte na tayo here. Banga male gametophyte. Gametophyte bang structure which can produce gametes male. A male produce pollen grain male. So male gametophyte is pollen grains. Here the female gametophyte is embryo sac because we will get XII inside this. So let us see. Artists of major Megasporogenesis zaucha dakha heikha functional megasporita jo. Here 3 degenerate. Remember, thumte kha hoilo lam mano dunjin sesuk le loma. Abana functional megaspore in kha bangan boldi. Repeated akha. Again, they'll form 2 cells. Here, they'll form 2 cells. Hilo le. Chanda will le draw le. Textbook adan chinay draw do le. They'll form 2 cells. So obviously it will be mitosis. Mitosis again. They'll form 1 apada 2 more cells. Yeah. Mitosis 3. Form randomly more cell stamping form 6 sitaja. Bana mitosis ma. That's simple. Bana Leon form so dumping form just from the one functional megaspore by repeated mitosis divisions 1, 2, 3. Akha baktiran dawa and form wa chile they'll form 4. Haika honki rearranged the engine, so we'll get our beautiful embryo sac. Thumte kha tungla ma paita ucha. Thumte paita u. Hai khadon paisuk khat paita u alai ki mang ki mu. Thum paita u thum tengan wa ya Jin swak tai boy. Simple mo. Hai khabang embryo sac. Development of female gametophyte. Yeah. Figure khabang nagyal do develop the development of female gametophyte or embryo sac. Malam. Functional megaspore apalakha repeated mitosis akha we form this. So we have to study its of this structure. What are these? Hai TCL te banga chima. Isil ngai do hai kha matured embryo sac ita. So this is how write the development or explain the process of how the female gametophyte is formed. Chile, hay apata na patayo mana. 1 cell apata 2, 2, 3, 4, 6, and 8. So embryo sac structure we draw the one like to draw so we have. Ay pin ka anal dawin mo, alian dawin ki draw that siya. Okay. embryonic structure hay teng thum bang mala lai ki ma om tigel bang anwaya thum te bang hay bang hay to hay bang athuma tung am pen kha we call it antipodals azat na function le omlom lom lo ma lu kham ngai lo atu alai ki ma kha we call it secondary nucleus le ki chi central cell le ki chi there is a large central cell, right? So we can call it central cell or secondary nucleus or two polar nuclei. I mean tamlu amo central cell ki chi ule secondary nucleus le ki chi le two polar nuclei. Bazaga cell khat hi changa kha central cell ki chi chu secondary nucleus two polar nuclei bangzaga ki tai chi le hai ti kha nuclei ni ki su so mai jo nuclei ni ki fuse ai changa kha mo two polar nuclei le chi the he alian pen kha anwai la malian pen kha bang ki chi XII hai kha hi lo mo hol hol pen no XII chi khong fertilization a apoi mo te kha abana anwai ya kha XII bodyguard gel kha synergies ki chi embryonic structure. So embryonic it is a female gametophyte. Male bangzaga female gametophyte actually because they'll produce female gametes. So what is the female gamete? We know XII, right? So what is the structure of embryonic? It consists of 3 antipodals, 2 polar nuclei, 1 XII and 2 synergids. Nachidiumam abana hai te kha ban mana function te kha textbook apadale abona kim vek male so hai kha it changa kha to apoi mo pen mom apoi mo pen chu en leuchin cell bangza sim deule cell bangza one a cell e ka chi chu central cell pen en leuchin nuclei pen hilawa mo a cell as a whole kha bangza chila 1 2 3 4 5 6 7 cell here right or wrong 7 cell de chu male embryonic to a nucleus sim leo chin bang sa di 1 2 3 4 5 6 7 8 male so we call it 8 nucleate 7 celled 8 nucleate so a typical angiosperm embryonic at maturity is 8 nucleate 7 celled 8 nucleate 7 celled so this is important 8 nucleate 7 cell ha structure te kha dang poimo lom lo in chin ha synergies te kha banga bang a function penu banga chile synergies te chu XII pe kha pote gunjina bana pollen tube hon pai changa kha he pollen tube hon pai dia male stigma pada male so when the pollen tube comes they'll enter through this synergids because they have this finger-like projections. Synergids have finger-like khutlim dan dewa mo finger-like projections ne unjin wai pen kha an hong tu mo ana welcome to male gametes te ana lut sak tu pen kha synergids chin wai jaga kha synergids te kha they are important. They have these thickenings known as filiform apparatus. Synergids have thickenings. You can draw like these finger-like projections. Imagine. So these are thickenings. Alright. We call it synergid has filiform bang FILLI filiform apparatus. DATUS apparatus. DUS apparatus, filiform apparatus. So it has synergies function in my engine. Antibodal statue, they don't have any particular function. Polar nuclei, we'll see polar nuclei in XII, they are going to help us in the fertilization. And embryonic at maturity is 8 nucleate, 7 celled. Mala. Like her, female gametophyte. Jen hai kha to kha XII kha the dia chu XII to two synergids kha ti gom changa kha hai kha XII to bodyguard te gom changa kha we call it egg apparatus azak chu apparatus anaset na sem khom ki we changa kha aya egg apparatus ke chin abana synergies kha banga importance kha they have what filiform apparatus kachima thickenings kha finger like projections mole they have filiform apparatus which help in guiding the pollen tubes mole guiding the pollen tubes into the synergid suma honlu thaile dinga kha ai kha a function synergids the function alu sak thei tu pen mo ahong tu pen kha mau so hetan henshin formation of the what is megasporogenesis the formation of megaspores from the megaspore mother cell mole so where do we get the megaspore mother cell because the nucellus they differentiate honki khento amunchin they form the megaspore mother cell nucellus ko it is in the central portion of the ovule and then out of the 4 megaspores 3 will degenerate one will become functional why functional megaspore benha banghon chidi it will develops into female gametophyte. What is the other name of female gametophyte? Embryo sac. And then we study the structure of embryo sac. Embryo sac is what? 7-celled 8 nucleate. Male atungla makha 3 antipodals alaya kha 2 polar nuclei or central cell. Anoila makha 1 egg cell which is protected by 2 synergids. Egg cell to 2 synergids ka egg apparatus ke chin synergids ka filiform apparatus ne engine finger like projections they help in guiding the pollen tubes into the synergids so that fertilization can take place. So now we are ready for the fertilization part. So this is it for today. Thank you.